Welcome to our webinar entitled Overcoming Purification Hurdles with MixedMode Chromatography, presented by BioCompare and sponsored by Bio-Rad. My name is Peter Fung. I'm the managing editor at BioCompare, and I'll be your moderator for the presentation. Before we begin, I'd like to share a few points with our listeners that this event will hold a live question and answer session at the end of the presentation. You can submit a question at any time during the presentation using the Q&A box located on your screen. And the resource box located on your screen contains downloadable documents pertaining to this webinar. Now allow me to introduce today's presenter. Dr. Mark Snyder is the manager of Process R&D Applications Group within the Process Chromatography Division of Bio-Rad Laboratories. Dr. Snyder received his PhD in biochemistry at the University of California, Berkeley under Dr. Daniel Koshland, Jr., and did his postdoctoral work in molecular biology with Dr. J. Michael Bishop at UCSF. He spent 5 years at SCIOS, then California Biotechnology, working on basic fibroblast growth factor cloning and purification, followed by 4 years as a manager of process development at Soma. Prior to coming to Bio-Rad, Dr. Snyder spent 13 years at Bayer Healthcare, where he was the Associate Director of Process Sciences in charge of the Purification Process Development Group. He led the team that developed Bayer's current generation recombinant factor VIII purification process and transferred it into manufacturing. His team also successfully developed and implemented a variety of Phase 1 and Phase 2 purification processes for other protein therapeutics, some of which are currently in late-stage clinical trials. Dr. Snyder has also played significant roles in process improvements, validation, troubleshooting, and optimization, and he's also been involved in viral clearance studies. He has authored licensed application CMC sections and inspection responses, and his work has been published in numerous peer-reviewed journals and various oral presentations. Welcome, Dr. Snyder.
Well, thank you very much, Peter, for that kind introduction. I'm excited today to talk to everyone who's listening about two different uses for mixed-mode resins. One is going to be purification of an adenovirus, and the other is going to be purification of a bacterial surface protein. So without any further ado, let's get started. As I said, the first is going to be purification of a virus. We're going to talk about purification of adenovirus. We chose adenovirus because, as many of you know, it's already in use as an effective tool for the transfer of genetic material. It's the most widely used vector for gene therapy. In fact, if you do a survey of the literature, you find that it's employed in about 25% of all gene therapy trials to date. As of 2014, it's the vector of choice in almost 500 clinical trials, in fact, and that number obviously has grown in the last year and a half. One reason why people like to use it is because it can accommodate a very large amount of genetic material, up to 37 kilobases of foreign DNA or non-adeno DNA. It has a very high infection efficiency. It can infect a wide variety of cell types, including both dividing and non-dividing cells, which is crucial for some gene therapy applications. The genome rarely integrates into the host chromosome, which is something that people prefer in a transfer vector, and the current production systems can generate very high titers. Now, there are a couple of challenges that adenovirus poses to downstream processing. One is the large size. It's about 165 megadaltons in size and has a diameter of about 0.1 microns. And I've shown on this slide a picture of what adenovirus looks like in the middle, and also in panel B. It's not generally stable to low pH unless it has a high salt content in the buffer. And there's also over 2,700 subunits, which means that there are lots of charge variants. Now, in the bottom right-hand panel, I've shown the major proteins that one sees on an electrophoresis gel of the adenovirus, and there are basically 5: the hexon, the penton, core 5, hexon 6, and core 6 proteins. And those will be the ones that we're going to focus on today. So what's Bio-Rad's approach to trying to use a mixed-mode resin to make a simple purification process? Well, we screened a total of 5 resins, which I'll go through in detail in a minute. We developed a 2-column, rather efficient process, and I'll go through the data on the efficiency also. We found that the process is readily scalable, it's very simple, It's very rapid. And most importantly, the process yields an active concentrated virus product that has the purity in terms of host cell protein levels and DNA contamination comparable to existing clinical grade products. So, material summary. First, we use a virus called Ad5E1 plus GFP. It is serotype 5. The E1A gene has been replaced with green fluorescent protein so that we can monitor the virus easily during chromatography. We grow it on HEK293 cells in DMEM and 2% fetal bovine serum. The harvesting is done by first treating the cells with a permeation agent, which is proprietary. Cells are then removed by filtration. And the supernatant is treated with turbonuclease. Now I'll have a bit more to say about turbonuclease at the very end of this presentation. Focus forming units were assayed via serial dilution and counting of the resulting plaques. The transgene expression, as I alluded to a minute ago, was monitored by fluorescence spectroscopy, and as you look at the chromatograms on the following slides, this will be shown by the blue shaded areas. Gel electrophoresis was performed using Bio-Rad's criterion, tris hydrochloride 4 to 20% gradient gels, and they were then stained with BioSafe Kumasi. And DNA was determined by qPCR using primers to the 18S RNA gene. Screening was done using our Foresight columns, and I'm going to go through the buffer sequences for those in a bit of detail here. For Unisphere S and Nuvia S, our cation exchange resins, columns were equilibrated in sodium phosphate at pH 6.8, and the harvest was exchanged into this buffer prior to loading, and then any virus bound was simply eluted with a high salt concentration at pH 7.4. For Nuvia C', our MixedMode resin, column was equilibrated in 25 millimolar histidine at pH 6.5. The harvest was exchanged into this buffer before loading, and then the virus was eluted with stepwise increasing pH and sodium chloride buffers, and I'll go through these data in a minute. For Unisphere Q and Nuvia Q, our anion exchange resins, columns were equilibrated in tris sodium chloride at pH 8.1. Harvest was loaded directly onto the columns, and the virus was eluted with stepwise increasing sodium chloride concentrations. So the results of the screening of these resins are shown in this slide. What you see is the list of resins on the left, followed by an indication of whether we found virus in the flow-through, wash, or in the eluate fractions, and a few notes. So what you see first is that for the cation exchange resins, Unisphere S and Nuvia S, we found virus both in the flow-through fractions and in the eluate, and we decided based upon these data that these resins would be not well suited for virus purification, either in bind-dilute or in flow-through mode. By contrast, Nuvia C' bound all of the virus, and it seemed to be eluted quite well in the presence of some sodium chloride. So what we thought was that probably we would require some dilution of this virus before loading it onto the column, because as many of you know, the conductivity of tissue culture fluid is often around 125 to 175 millimolar sodium chloride. Unisphere Q and Nuvia Q, our anion exchange resins, did not bound the virus quantitatively, and it quantitatively came off in the eluate. And we also thought that perhaps this could be considered for direct mass capture. But at the end of the day, we selected Nuvia C' for the initial capture step, and there were two reasons. The first was that if you compared the results between C', Unisphere Q, and Nuvia Q, C' gave the best clearance of feed stream contaminants. And also we thought that there would be a minimal manipulation required of a C' eluate prior to loading onto Q because of the ability of some of these Q resins to bind in very high salt. And I'll go through these data in more detail in a minute. So what I'm going to show you in this slide is some iterative development of the Nuvia C' capture step. In the top left-hand panel, panel number 1, you see the chromatogram which resulted when the feed was diluted 1:1 with 75 millimolar histidine at pH 6, applied to the column and washed with that buffer. And then the column was eluted with step elutions of tris containing 0, 125, 500, or 1,000 millimolar sodium chloride at pH 8. What we can see from this chromatogram is first, a significant part of the virus flowed through the column, meaning it did not bind under the conditions. And second, that some virus did elute, and from other experiments we know that the elution really is around 500 millimolar sodium chloride. This led to the development of the second experiment, where instead of diluting the feed 1:1, we now diluted it 1:3 to lower the conductivity. This resulted in all of the virus now binding to the column. The elution buffer, again based upon some experiments that I'm not going to show, was optimized to 75 millimolar tris, pH 8.5, plus 525 millimolar sodium chloride. What I want you to notice are several things. First, there is still a small amount of virus which did not bind tightly to the column. Second, the elution peak is actually split into two. Now, if you carefully compare the elution peak from panel 1 and panel 2, you don't see the split elution very clearly in any event in panel 1. We're not sure what the reason for this is, but we decided to leave it for this purification process because it might become relevant down the road to somebody who's actually using this adenovirus serotype for further work. Panel 3 then shows the last iterative change. Instead of washing the column with 75 millimolar histidine at pH 6, we wash the column with 25 millimolar histidine at pH 6. And now you see all the virus is bound tightly to the column and all of it elutes in the 525 millimolar tris elution step. And what you see then is the final process as I just described. Equilibration of the column in 25 millimolar histidine, loading of the sample diluted 1:3 with this buffer, a wash, followed by elution with the tris-salt combination. These are the results of a chromatogram in a second experiment just to make sure that we had things right. And again, what you see in the top panel is the complete chromatogram from start to finish. In the bottom, what you see is an enlargement of the elution section. And you'll notice that there are two peaks coming out in the elution. Both of them contained active virus. We couldn't find any significant difference between the two using the limited analytics that we had, but we collected both peaks. Now I'm going to move on to the polishing resin. As I indicated, it looked like both ViaQ and UniSphere-Q would be suitable in a bind-elute step. So what we did to compare the two is we loaded both with a virus preparation and then did step elutions. What you see in this panel is that ViaQ retained the virus longer than UniSphere-Q did as we increased the concentration of sodium chloride. So we selected ViaQ on the basis that coming off the ViaC prime column at 525 millimolar sodium chloride, ViaQ would be better suited for direct capture than would UniSphere-Q. So in this panel, I'm going to show you just a bit of development of the ViaQ polishing step. What you see is are two chromatograms showing virus loading, followed by washes with two different buffers. In the top panel, there's a wash with 75 millimolar histidine, 400 millimolar sodium chloride at pH 8. And what you see on the right is an SDS page gel of the various fractions. Panel C is the elution of the virus material at a higher salt concentration. And you'll notice I've indicated the 5 readily apparent virus bands that I showed in the very first slide by red asterisks. But in addition, there's a band that's above the topmost virus protein. And we suspected that that was a host cell protein contaminant. So what we did to mitigate this is we increased the wash concentration from 400 millimolar sodium chloride to 440 millimolar sodium chloride. Now that doesn't make a visible impact on the chromatogram as you might expect, but if you notice on the lower SDS page chromatogram, that did result in a shift of that band from the eluate into the wash fraction, which is what we were looking for. So the final polish conditions, we're equilibrating the column in histidine at pH 8, loading the C' eluate 1:1 with this buffer, diluted 1:1 with this buffer, washing the column with an intermediate salt concentration, then the final salt concentration wash of 440 millimolar sodium chloride, finally eluting with 75 millimolar tris, 1 millimolar salt at pH 7.5. And again, this slide shows the final chromatogram, the load, flow through, the pre-elution peak containing that suspected host cell protein, and the product collection. And you'll notice this time that the product eluted is a single peak rather than a split peak. Okay, so these are some scale-ups, 5-fold scale-up to a 5 mil column using the final process. And again, I've shown the chromatograms. On the left is the ViaC prime chromatogram, again with this split peak eluate. On the right is the ViaQ chromatogram with the nice sharp single peak eluate. So we did some analysis to see how we did. On the left-hand side, you see the results of some intermediate fractions and the final fraction coming off of the Q polishing step. The final fraction I'm just going to focus on is in lane 7, and really what you see are just the 5 proteins, or predominantly just the 5 proteins, that I indicated from the first couple of slides are the major adenoproteins that one can see on a gel. And there's very little contaminating host cell protein. We did a few experiments to look at what the level of contaminants might be, and also at what the overall yield from this process was. I presented this on the table on the right. First thing to notice is if you look at the total virus, what you see is that there was about a 55% yield from bulk harvest through the final Q eluate of virus particles. And I should point out, it's not shown on this slide, but these were also active particles. So these particles were, we recovered mass as well as that amount of activity. And in the right 2 columns, what you see is the DNA and host cell protein levels per 10 to the 10th virus particles. The DNA was below detection, and the host cell protein was quite reasonable at 2 nanograms per 10 to the 10th virus particles. So the conclusions from these sets of experiments were, first, we developed a simple 2-column workflow for adenovirus, which we designed and tested. Purity and host cell DNA levels were suitable for clinical products. If you do a survey of the levels of these contaminants throughout a whole variety of clinical processes, you see that it's very reasonable. The process is readily scalable. All the buffer components are cGMP compatible. Now, I did mention that I was going to have a note about turbonuclease. Turbonuclease is not yet cGMP compatible, but other nucleases, for example benzonase, can be substituted with equal efficiency in this process. And the combination of mixed mode and anion exchange chromatography produce highly pure adenovirus with high yield and low impurities. So now I'm going to move on to a second use for mixed mode chromatography, the purification of a bacterial surface antigen. So what were the issues when we started this project? Previous processes to purify this antigen were very long, they were time-consuming, and they had significant mass loss. The yield was on the order of 10 to 15%. In addition, there was a huge loss of activity across the purification in terms of the residual antigenicity of these proteins. Also, there are a variety of serotypes, and each had a different published purification protocol. So what we were hoping to do was to simplify things and perhaps come out with a platform process for these serotypes. So the first thing we did is we chose one serotype, which I'll show you later on. We did a DOE design of experiment to optimize the elution condition. Binding was in 50 millimolar acetate at pH 5.5, and in this table, what I've shown are the elution conditions that we used. So we had a center point of pH 7, 250 millimolar sodium chloride, and we went to both pH 6 and 8 for the elution pH, as well as a salt concentration of 0 and 500 millimolar. And the response surfaces that we were looking at were purity and yield. So on this next slide, what I've shown is an analysis of the DOE data. First of all, what you see from the top panel is that in terms of product purity, both elution pH and sodium chloride were important. And you can see that from the t-values shown on the right-hand column. But in terms of product yield, which is shown in the bottom, neither elution pH nor sodium chloride concentrations seem to be particularly important. Again, this is reflected in the t-values and the t-ratios for these parameters. If you graph the analysis of the purity data, what you see is something like this, where both lower pH and lower sodium chloride gave a pure product. So we use this data to design a temporary or a trial elution scheme. What we did was loaded the material onto the column at pH 5.5 out of the cell extract. Then we eluted with a gradient from pH 5.5 to pH 7.8, going from acetate to phosphate. We took various fractions and analyzed them, and I'm going to show you that on the next slide. This is the elution chromatogram that we achieved. So here we see both SDS page analyses of the individual fractions from the previous slide, as well as some other analyses. What you see from the reducing SDS page is that we got this bacterial surface antigen in fractions 11 through 16. And when we did offline pH measurements of these fractions, we get the values shown in the second column of the table. Purity analyses by densitometry showed that the purity was rather high in going from fraction 11 up through 14, and then dropped at fraction 15. So on the basis of these results, we converted this gradient elution to a step elution using 50 millimolar mess at pH 6.2. What we got is shown in this slide. Here you see the step elution. There's a significant flow-through peak, followed by the step elution with a relatively nice peak and some degree of tailing. When we analyze this fraction, you see that the pooled fractions give a good recovery of the surface antigen, not very much lost in the tailing fractions that we decided not to take. And the pH at this pool was 6.2, as we expected. So in this slide, I'm going to show you the development of the polishing step on ViaQ. We already knew that these surface antigens would flow through anion exchange columns. So what we did was take the eluate from the ViaC prime column, which was at pH 6.2, and altered the pH to either 6.0 or 6.8, and applied then those loads to ViaQ columns equilibrated at those two pH values. All the bacterial surface protein flowed through, and we then stripped the column with 1.5 molar sodium chloride at a higher pH to see what remained on the column. What you see then in lanes 2 and 5 are comparisons of the two flow-throughs at pH 6.0 and 6.8. They look fairly similar, although one might argue from just these two lanes that the purity in lane 2 seems a bit higher than that in lane 5. If we then look at the material which remained on the column, namely lanes 3 and 6, we see that this is actually confirmed because it appears that the impurity bands in lane 3 are slightly darker than they are in lane 6. So as a result of this, we decided to use a pH 6 rather than pH 6.8 because it did appear to offer a slightly higher purity. So again, these are the results of a slightly larger scale process run with this method. It was simply 50 millimolar mess at pH 6.2 run in flow-through mode. And what you see is a highly pure protein, as shown by the monomer peak, but in addition there appeared to be some small amount of dimer. We decided to have a bit closer look at this, so we ran both reducing and non-reducing gels, and what you see are a series of proteins which appear more in the non-reduced lane than in the reduced lane. There's some sort of oligomers or perhaps slightly altered conformations even in SDS page gels. We're not sure exactly what they are. This will be left for some future work. So this was what we got for this particular antigen, for this particular serotype, which we'll call type 1. There were 3 other serotypes, types 2, 3, and 4. We were looking for a platform approach to these other serotypes. And what we found is, in fact, a fairly decent platform. Types 2 and 3, we could bind at 50 millimolar acetate pH 5.5. Type 4 required binding at a slightly lower pH, pH 5, but for all types, we were able to elute them with a 0 to 20 millimolar sodium phosphate gradient at pH 7.8. And the Q polishing for all the types was the same, 50 millimolar mess at pH 6.2, run in flow-through mode. These are summary gels of types 2, 3, and 4. And again, what you see in lanes 3 for all of them, the final ViaQ flow-through, again, very highly pure fractions. So in summary, we developed a rapid 2-step purification for this surface protein. The overall mass yield, although I haven't shown you the quantitation, was greater than 50%, and of this remaining 50%, the activity was 100%. Finally, all serotypes show a similar purification properties. So we believe not only have we developed a simpler, higher yield process for this surface antigen, but we're also well on the way to developing a platform process for the various serotypes. So I want to thank you all very much for listening, and I will now open the lines for questions. Thanks very much.
Thank you very much, Mark, for a great presentation. We'll now begin the question and answer portion of our webinar. So thank you, everyone, for submitting your questions here. So to start things off, the first question we have is, when would mixed mode chromatography be beneficial over traditional?
Right. So the advantage of mixed mode chromatography is there are actually two advantages that might make you choose it over traditional. One is that the separation mechanism is different than the type of chromatography that only employs one mode of adsorption-desorption. So you get different selectivities. So you would employ it when the traditional methods just aren't working well enough for you. Second advantage that mixed mode has over some other resins is that you can bind in fairly high salt. So if you're looking to load a fermentation harvest directly onto a resin without having to do any dilution in any event, you might look at mixed mode.
Okay, great. I've got another question here is, can I load fermentation harvest onto ViaC Prime directly?
Right. So again, the answer to that is yeah, you probably can. We've looked at the binding capacity, for instance, of ViaC Prime for Q and IgG, and what we find is that the binding capacity is fairly constant up to 30 millisiemens per centimeter, which is well above where you typically find fermentation harvests.
Okay, great. I've got another question here is, how can you decide what mixed mode resin to use among all the available mixed mode resins?
Yeah, that's a great question, and the answer is really good news for process developers. It's going to keep you in business. The answer is, besides broadly separating them on the basis of charge, in other words, if you have a positively charged protein, you're going to want to pick a mixed mode ligand that has a negatively charged feature on it. Beyond that simple discrimination, there's no way to tell which one is going to work better than another one, so you just have to try all of them. And Steve Kramer from RPI on the east coast of the U.S. has published a number of papers on this, and I direct anyone who has more interest in looking at some of the details to some of his papers.
Great. Okay, Mark, I've got another question here is, what about mobile phase modifiers? Do they affect ViaC Prime chromatography?
Yeah, that's another great question. The answer is absolutely they do. And you can think of pretty much any mobile phase modifier you want. And the list offhand that comes to mind is arginine, urea, guanidine, glycerol, ethylene glycol. I'm sure I'm missing a couple common ones. They all affect proteins differentially on C prime, meaning that they will affect a particular protein in different ways on C prime itself, and if you compare C prime to other mixed mode resins, they will differentially affect the behavior of a particular protein on C prime compared to that same protein on another resin. So again, if you're looking to do some really fine separations and C prime itself isn't giving you what you need, you might try the suite of mobile phase modifiers that are probably in everybody's toolbox.
Okay, and let me see, I've got another question here is, what else has ViaC Prime been used to purify?
So we and our customers have looked at a whole variety of proteins, and really the simple answer is it spans the gamut of every kind of protein class that you can think of. I can't give a lot of specifics, but there are structural proteins and enzymes, and immune-based proteins, and.
Okay, and I've got another question here. It's similar to the previous one. Is how does this resin compare to other mixed mode resins? Maybe some thoughts you can share?
Well, again, your protein might flow through one mixed mode and bind very tightly to another one. And let's assume for the moment that they're both hydrophobic cation exchange type resins. So there's no way of predicting. You just have to try it. I hesitate to directly compare the results that our customers have given us head-to-head with other resins because I don't believe that's a fair thing to do because somebody else can get exactly the opposite result. So I think I'm just going to leave it at that.
Okay, Mark. And those are the questions we've had in. Are there any additional comments you'd like to share with our audience and listening?
Well, I'll share a personal comment, which is, education of our customers and answering questions is what I do for a living, and I really enjoy doing it. So if any of you have any questions that come up after this webinar, you can either contact your local Bio-Rad representative who would be happy to help, or you can also, if you want, contact me directly. My email is mark_Snyder@bio-rad.com.
Great.
And thanks, everybody who joined into this webinar.
Wonderful, Mark. And we want to thank you for taking the time to share your knowledge with us in a great presentation. And we also want to offer a very special thanks to Bio-Rad for sponsoring today's event. So please keep a lookout for an email containing a link to the on-demand version of this webinar. And thank you, everyone, for attending, and have a great rest of your day.
Thanks, you too, and thanks, everybody, again.